So we're gonna go ahead and begin the panel. The focus of this session is to explore the potential of cell therapies in central nervous system disorders, including stroke, spinal cord injury, and age-related macular degeneration. So there's a lot of coverage here, that we have to go through, and so I apologize to the speakers right off the bat if we do not go through your illustrious histories, but you'll see that these are all top key opinion leaders in their various sectors, and hopefully that'll come out as well as they're speaking. At the end of the session, if you as an audience can leave with an understanding of what the disorder is and what the unmet need is, if you can leave with an understanding of how cell therapies have worked historically and sort of where they're going, and if you can leave with an understanding of what clinicians want to see from the ongoing clinical trials that are going right now, I would consider this this panel a success. So we're gonna jump right into it, and start a discussion with Dr. Savitz and Dr. Wechsler, both who are neurologists by training and have been involved in the stroke space for decades. Doctors, if you could let us know what is the the current standard of care in ischemic stroke, and I guess what are the limitations that you see that potentially could be addressed with cell therapies? I'll let either one start off.
Since I'm the older of the two of us here, I'll start off. I've been in the stroke field a few years longer. I go back about 30 years, really, and I can tell you that when I first started out in stroke, I learned two things. One was that the brain doesn't fix itself, that there is no recovery in the brain. And the other is that the only thing that we can do for a stroke is to prevent it. And once the stroke has happened, it's over, and there's nothing we can do. What has happened over 30 years is that obviously both those things are wrong. The brain is an active recovery organ, and there are many things that we can do for stroke besides prevent them. When I first started in stroke, obviously the emphasis was on prevention of stroke. Things like aspirin were very important to us. Then around in the mid-1990s, we developed treatments for acute stroke, things like intravenous TPA, subsequently intraarterial therapy. And that really became the focus of much of the stroke community, how to intervene in the setting of a stroke and prevent the damage that otherwise is going to occur if you don't do those sorts of acute therapies. Certainly prevention has a huge impact on stroke. Acute therapy has as much energy as we put into it in the stroke field, has a very small impact. Only a small percentage of patients get into the hospital in time for these acute therapies. And so what we're left with is a huge population of stroke patients who, despite our best efforts, we are not able to prevent their stroke. Despite the fact that we have these acute therapies, they're not able to access those acute therapies for timing or whatever, and they're left with significant neurological deficits after a stroke. And that's a huge population of patients. In the United States, probably 4, 6, 8 million people living with the deficits of stroke, many of them severely disabling deficits. And what we have to offer those patients is virtually nothing right now. We do physical therapy, we do occupational therapy, we do speech therapy, and sure, that does do something, and we are currently obviously refining that to try to do more. But most patients plateau in terms of the recovery of stroke after 3 months or 6 months. And from then on, they are left with whatever deficits are still residual at that point. And so from my point of view as a stroke physician who's been in this field for many years, we are desperately in need of something to offer these patients. This is where we need to devote our energy in the stroke field for the next decade, for the next many decades. So that we can approach this problem and help people who have these really devastating deficits.
Maybe Dr. Savitz can also, jump in and talk about how cell therapies may be able to address some of these deficits. And probably, just as important for both of you, what are you seeing right now that suggests that it could?
Yeah. So following up on Larry's point, a big part of, I would say, the attractiveness of cell therapies for stroke is, just as Larry said, you have right now, in terms of standard of care, if you want to be able to treat somebody with standard of care therapies, you have to treat them really quickly after the onset of stroke. So where there is opportunity is that what we now know is that after a stroke has occurred, there's a rather robust inflammatory or immune response which occurs after stroke, and there's also repair mechanisms that are occurring after stroke, that are occurring within the brain. There is an active attempt to try, let's say, neurogenesis, angiogenesis, even synaptogenesis, is starting to occur in the first few days or even weeks and even months after stroke. And so cell therapies, many different types of cell therapies have been found to actually boost or upregulate the repair mechanisms that are occurring in the brain. Some types of cell therapies have also been found to modulate the inflammatory response very, very interestingly and selectively in ways that may actually help to facilitate recovery after stroke. So, where there's great opportunity, and we've had 10 years now worth of animal model literature in stroke models, where there's been opportunity is to actually see that cells, whether they are from bone marrow, umbilical cord, various other types of tissues, a lot of the, I would say, non-neural cell types, many of non-neural cell type therapies have been found to upregulate repair mechanisms and to impact on the inflammatory response. And this is data that's been generated, I would say, over several years. And there are windows of probably opportunities for cell therapies to exert their effects in the days and maybe even weeks after the onset of stroke. And then where the literature is really going beyond this is we're finding through our partnerships, for example, with industry groups like Athersys, that the cells, like for instance, their bone marrow-derived stem cells, may actually be having impact not even within the brain directly, but through organs in the body like the spleen. And we're finding that other organs, for instance, like the spleen and maybe even the lungs, could be participating in the inflammatory response after stroke. And that these organs may actually be very important to have an impact on them to facilitate recovery. So it's a really intriguing time to think about how cell therapies could be used. And that's just for the first few weeks after stroke. I haven't even talked about even what we might be thinking about for chronic stroke.
So before we go to chronic, and maybe back to Dr. Wechsler, can you talk a little bit about your clinical experience? Because, as all of us, especially investors in the audience, know, we've cured mice 1,000 times over of cancer and every other, injury there is there. But the translation from preclinical to clinical is obviously of paramount importance, and especially trying to time an investment. Where are we in the clinic, and what has been your experience so far in the clinic? And where I'd like to go with this, or end this little section, is, what are the endpoints we should be looking for? Should we be looking for classic endpoints of walking time and things along those lines, or should we be redefining how we look at this group of therapies?
So obviously really important questions, and Sean may have a different view of this than I do, but I think stroke is one area where it's very difficult to translate from animal models to human stroke. The gap between a stroke in a mouse or a rat to a stroke in a human is massive, and they're very, very different. So I think that's been a problem with stroke in general, and we're seeing it again in this area of regenerative medicine. So my experience has, I've been doing studies, human studies with cell therapy now going back to 2000. We were involved with Latent Bioscience and their cells and several studies in between. But of course, mostly we're focused on safety issues and feasibility issues. On the efficacy side, we face a number of difficult problems. The appropriate endpoints, I think, to your point, are not the ones that we traditionally use in our stroke trials to date. As I said, the stroke field has been focused on this acute stroke area, and we're looking for huge, big jumps in ultimate recovery. we're probably not going to see that in regenerative medicine and cell therapy, at least not right now. And so we've got to, I think, come up with new endpoints that are more relevant to the things we're doing. If I can take someone who can't walk because their leg's too weak and make their legs strong enough that they can walk, that's a huge change for that person in their quality of life. If I can make their hand work that couldn't grasp anything, but now they can hold something, that's a huge difference in their quality of life, and that may not be measured in any of the scales that we have to date. So I'm completely with you. I think we need to redo the endpoints. We need to rethink how we're looking at this and somehow convince the FDA, and I think that's the hard part, convince the FDA that our approach, new approaches to this are valuable enough that it can lead to approval, because if we ultimately don't get approval from the FDA, then you guys don't have a product, and then we don't have a treatment for our patients. So we've got to work on that end as well.
Dr. Savitz?
Yeah, I would agree. I think that the endpoints that have been used to look for efficacy in acute stroke studies like TPA may not necessarily really be the best ones for regenerative therapies in stroke, where we're looking at enrolling patients that are days or weeks afterwards. So modified Rankin scale is the one that's being used in stroke studies, and that's a 6-point scale. And it could be crude in the sense to be able to pick up smaller changes, let's say, in recovery. So there are other modalities that could be looked at to look at hand function, or say arm function or leg function, or just motor function in general. It's like Fugl-Meyer score could be one example. And we do know that CBER has been open to other endpoints in addition to the more global disability scores. But the companies that have gotten these trials off the ground recently, like Athersys and Aldegen, are aware of these limitations, at least from my vantage point in seeing that, and they have come up with more creative ways or more sophisticated ways to use traditional endpoint scales to maybe see treatment effects, because it may be possible to consider in the first few days after the stroke, if you have equal percentages of patients with certain disabilities, then you could see where they're going to be 90 days out. And if you had just a shift in some of these disability scores by 1 or 2 points on our traditional disability scores, that might make a difference. In the past with TPA, we were going for a home run, a complete cure, where you have just minimal symptoms, but you're completely independent. But in the studies that these companies have now designed, it may be possible to look for a treatment effect in which you just see them shifting down on their disability score, just getting a bit better, and shifting like that might actually be one way to try to find a treatment effect.
So sorry.
I would just add one other thing. I think that something else we ought to focus on is perhaps spending more time on early phase 2 studies and later phase 2 studies before we go into phase 3. I think there's always the rush, particularly in industry, to get to phase 3 to prove the efficacy of your product. But I think if we spend more time looking at phase 2 studies, surrogate endpoints, imaging endpoints, infarct size, things like that can give us a better indication of clinical efficacy, we're going to be much smarter about going to phase 3, designing the trials better, and knowing what is going to have a better chance of succeeding in that arena.
Now, one of the things you as the audience will find out is that each one of these indications we could spend an entire panel on. But just to wrap up just this section, what are you seeing in the clinic right now? So, for example, I understand in the preclinical side what we're seeing. What are the effects that you're seeing that has you excited?
Well, I think, and from the clinical side, I'm really excited over the fact that there actually is now buy-in on the industry side to actually do trials on stroke. Okay. I think that we have to start there, quite honestly, because we haven't seen a lot of industry that's come into stroke in a while since the neuroprotection days. And I think that there's excitement because this is a completely different way of looking at therapies for stroke. And the idea that there are maybe several windows of opportunity within days or weeks after the onset of stroke. So from a clinical trial perspective, I can't say that we've seen any treatment effects yet because we're still in early stage clinical trial testing. We're still in the phase 1, phase 2 trials. The fact that we even have phase 2 randomized studies with 2 arms I think is actually an advancement for the stroke field overall. The fact that we have a control and placebo-controlled groups, and we're going to have data from trials that are going to be coming very soon, within a year or two, I think is really very exciting. I can only speak to our own work at UT in terms of a potential treatment effect, which is that, and I only say potential because we had designed a safety study looking at a patient's own autologous bone marrow cells given intravenously, and all we were able to do in a safety study was just to compare the patients up against control groups that were historically designed. They're coming from historical control groups, and we are seeing quite a difference between the groups, and that's data that's been presented, but that's only indirect, at this point in time. But it does give us a glimmer to give us an idea that cell therapies really may have a chance to work.
I can tell you that one difference between Sean's studies and our studies is that we are, dealing with chronic stroke, and that may be a disadvantage in terms of mechanism and so on. But there's an advantage when we're looking at efficacy, even though these are not designed for efficacy. But these are patients who are presumably past they've plateaued in terms of their recovery. In fact, in order to get into the study, they have to have 2 examinations a few weeks apart that show no change. But some of these patients are a year or 2 out from their stroke, and we don't expect them to be changing. So when we give the cell therapy, any change that does occur, you can perhaps think might be related to the cells, as opposed to patients who are still in the recovery phase, and it's hard to tell. And I will tell you that we have seen some patients who feel that they are better and actually have had some improvements in their examination. Patients have reported cognitive changes. They feel their memory stronger. They're able to do things they couldn't do before. And so these are encouraging. But we, as investigators, we understand that these are uncontrolled trials. There are no controls here. And these are patients who know what treatment they've gotten, and they come to these studies because they're very motivated to get better. So I don't have any idea whether it means anything. I think until we get to properly controlled trials, we wouldn't even attempt to make any conclusions. But at least we're not seeing people get worse clinically. That's a good thing. And we at least see some glimmers that make us think that perhaps there's something going on.
So let me switch gears then to industry. And Gil, you're CEO of Athersys, and one of the few companies, including Cytomedics, that's out there being a trailblazer in this space. Talk to us a little bit about what you've learned, what you're seeing from the models, and how this interaction with the FDA is going. Have you been successful in talking about how cell therapies may behave differently than other therapeutics that are being developed?
Absolutely. So the first thing that I think we all have to acknowledge and remember is that this is a big hill. Stroke is a pretty challenging condition. Just to kind of echo some of the points that Sean and Larry were making, it's an enormous burden from a public health care perspective. We've got about, according to the World Health Organization, we've got about 15 million people a year that suffer a stroke. About a third of those patients will die either right away or in close proximity to after when the stroke has occurred. About 50% of the remaining patients after the stroke has occurred are left with essentially permanent severe disability that affects them for the rest of their lives, and about the other 50% of the patients will live with moderate to virtually no disability, or disability that can be compensated for through physical therapy, occupational therapy, and the like. Despite the best efforts of a lot of pharmaceutical companies that have tried to develop neuroprotectant drugs over the past 15, 20 years, that's been an utter failure, and I think we have to acknowledge that. I think the primary difference between what we're doing with cell therapies like multi-stem and some of the other stem cell approaches that are being taken by Cytomedics and some of the other companies that are out there is that cells can do multiple things in parallel and can actually shift the body's healing responses in a way that a single agent therapy like a neuroprotective pharmaceutical drug just simply can't do. You've got multiple different things that are going on in the wake of a stroke, and cells are dynamic living entities that when they're administered can profoundly have an impact in terms of shifting how the response occurs. But I can tell you, having a board of directors that consists of a number of guys that spent their entire careers in big pharma, when you start talking about stroke, their initial response to that type of conversation is, "You've got to be kidding me." because they've all lived through stroke programs, and they're intensely skeptical about what it might be to take on a program in stroke. So the good news is that having a lot of skepticism about what the potential might be to develop a new therapy for an indication like stroke makes you work that much harder to really understand what's going on. And one of the things that we've spent literally years, a good part of the last decade, focusing on, as both Sean and Larry were mentioning, is it's not just enough to go into the prevailing animal models, as limited as they are, but those are the best things that we have, and show that you can actually see a good safety profile and a meaningful therapeutic effect that's reproducible in multiple independent labs and in numerous independently conducted studies. You've got to understand how you're actually achieving that therapeutic effect. And this is one of the things where I think the field of stem cells and regenerative medicine, where the progress has been tremendously exciting in the field of neurology, but also other areas that have been talked about in some of the other panels today, is that in contrast to where we were just a few years ago, where everybody was thinking about, well, maybe someday we can grow new neurons and we can actually transplant those neurons into the brain, and then we can just replace what's been lost due to a stroke or a traumatic brain injury, or in the case of a spinal cord injury, or other types of diseases, whether acute or chronic conditions. Now we know that cells can actually express a range of different factors that profoundly alter and enhance the body's healing processes in ways that are very powerful and also very durable. So as we were preparing to think about going into a clinical trial for stroke, the first thing we had to do was convince ourselves and convince our board regarding was, well, how is this therapy different from some of the classical approaches that have been tried? What are the various things that they're doing? And we learned some very surprising things along the way. Sean mentioned one of them earlier, which is these cells will actually go to organs like the spleen and influence, they'll shift the immune system away from that pro-inflammatory response that typically occurs after a stroke and actually exacerbates or amplifies the damage that's done, and in fact makes a lot of that damage permanent. And it'll shift it towards a reparative response where the inflammatory damage is minimized or muted in a very substantial way, and the body's natural desire or tendency to actually recover or repair itself is dramatically enhanced. So we've seen this across a range of different studies, not only in stroke, but other neurological conditions that we've been focused on. Others have seen very similar results. That's very comforting because we're all seeing a lot of the same types of biological phenomenon that is occurring, and then we start to see some signs that this is being translated into clinical activity. Well, that's when the field really gets exciting. And from an investor's perspective, you can ask yourself some pretty basic questions. How big is the opportunity? Right? We've conservatively estimated, and other independent analysts have done the same kind of math. I mean, stroke is probably a $15 to $20 billion a year opportunity, and that's if you're using very, very conservative estimates.
So let me just jump in, because before we get to opportunity, which I think everyone in the audience knows these are multi-billion-dollar opportunities, I think one key point that I hope the audience walks away with is, one of the mechanisms of action, or something you should be thinking about that's a commonality between a lot of these companies, is this concept of restoration of balance, and that applies to various indications. And just to keep everything going on time, I just want to switch gears very quickly to Dr. Del Priore, who's involved in macular degeneration. I'd love to ask you to start off this section understanding, what's the unmet need there? It seems like there are a lot of biologics that are available that are addressing the problem quite well. What is the unmet need that you think cell therapies may be able to help with?
So I actually think that there's been tremendous advancement in the field of macular degeneration, and it's been interesting because I think that the investment community knows about as much about this as most ophthalmologists do, because these have really been blockbuster products. With Eylea for Regeneron and with Lucentis for Genentech. But those are geared toward a specific subtype of macular degeneration called wet macular degeneration, where patients develop bleeding in the back part of the eye. And there actually still is quite an unmet need, because even with those products, which are actually been enormous clinical successes and enormous financial successes for everybody, there are still patients who continue to lose vision, and we could spend the whole day sort of talking about what other products are coming down the pipeline just to help with that particular disease. I think for stem cell therapy and for cell-based therapies, that the main application is going to be for a form of macular degeneration that we really have no treatments for right now, and that's the dry form of macular degeneration. The etiology, why it occurs, how it occurs, is a little bit less clearly understood, but essentially patients start to lose cells in the center of the retina and develop something called geographic atrophy. Geographic means it sort of develops in a patch, atrophy means that there's thinning or loss of cells in the retina. And at the moment, that's actually a huge unmet need. There really are no products that are FDA approved or even being used off-label to sort of stop the progression of this disease. And I think it's a wide open space, actually.
And so when we talk about cell therapies, let's say in dry AMD, what are the endpoints that we should be concentrating on? Is it the classic endpoints that you would look at with Lucentis or Eylea, or again, like in the stroke field and in other fields, are we looking at something more paradigm shifting?
I think the endpoints are. I think the measures will be different, but I expect that the endpoints and the efficacies will be different as well. So what happens with the wet macular degeneration products is that you can get significant improvement in vision just because the drugs essentially stop bleeding, and some of the loss of vision is really because of blood or fluid underneath the retina. And as soon as you stop the bleeding, the vision really can improve quite dramatically. Now, for dry macular degeneration, I think that there's two potential things. First of all, this is a rapidly progressive disease, well, relatively rapid. Over the course of a couple of years, patients can lose a tremendous amount of vision. So I think you'll see products that will be testing two endpoints. One endpoint will be just cessation of vision loss, where the vision just stays stable and doesn't continue to deteriorate. But then for cell-based therapy, I think that the ultimate bar that they're going to have to cross, hopefully, will be visual improvement in these individuals.
And I think, from when we had talked, you had talked about the history of this space and the fact that both adult and fetal cells have been tested. What, are they moving forward? What were the challenges, with those cells?
So there have been small clinical trials. Most of them have been done with the wet form of macular degeneration, but there are a few studies that have been done on the dry type of macular degeneration using transplantation of tissues from a donor into a recipient. And there you run into all sorts of problems. With adult cells, the main problems you run into essentially are graft rejection, and the second thing is the irreproducibility, if you will, of the particular lot of cells that you're using to actually try and repair the tissue damage. So the studies with dry macular degeneration have actually been very few. There were a few patients who had patch transplants of RPE, probably going about 10 years ago, for the dry form of macular degeneration, both with adult and fetal cells. And I think the results of those studies, a lot of the patients were not immune suppressed, and the cells, you would expect them to be rejected immunologically. And the results of those studies were equivocal at best.
So let me switch very quickly to Gary Rabin, who's the CEO of Advanced Cell Technology, who's developing, obviously, cells in macular degeneration. I had asked Dr. Del Priore to kind of talk about your results, but he was a little bit reluctant, until he got sort of a full update. So I'd love to give you, a couple of minutes to just talk about where you are right now and what is the latest clinical data from your ongoing studies in both Stargardt's and dry AMD, and then also address the two issues that Dr. Del Priore brought up: this idea of graft rejection and irreproducibility, thank you, of cells.
Okay. First of all, we are, because we're a publicly traded company, and because the information about patients, we obviously have restrictions as to what we can discuss. But we have spoken generally about the results that we've seen here. And in fact, the results of the first two patients were actually published in The Lancet, and we're now working on an update paper related to those two patients as well. Generally, we've seen a variety of different visual acuity changes in patients. And it really has run the gamut from patients that have picked up letters, lines on an eye chart to no changes in the Snellen visual acuity, but seeing changes in seeing changes in color, contrast, clarity, night vision, things like that. So one thing that's important to understand is that we've been treating patients that had hand motion or visual acuity of 2800 or 2400. These, as Dr. Del Priore described, These are patients that have extensive atrophy. They've lost a significant amount, not only of their RPE cells, but of their photoreceptors as well. And so when you get to this degree of atrophy in the photoreceptor cells, there's not much there to rescue in terms of visual acuity. When the photoreceptors are gone, they're gone. Now, we have separately made photoreceptors and photoreceptor progenitors, and we've seen some pretty remarkable animal studies, both by directly injecting them, I don't know what that's trying to tell me, both by trying to inject them directly into the retina, as well as paracrine effects by, via tail injection when we were doing tox studies. So, we see a lot of promise here. I think we've just begun a new cohort. After treating 18 patients, the FDA permitted us to begin treating much better visual acuity patients. So now we're treating patients, we're going to treat a total of 8 patients in the U.S. that have visual acuity of around 2100. So these are people that don't have their driver's licenses, but you could walk around with them and they can carry on. And the amount of atrophy, especially within the center field of vision, is much less than we've seen in the patients that we've been treating with hand motion in 2800 and 2400 vision. So I think that we'll see a lot more in terms of the effects of these cells on visual acuity as we begin to treat these patients, the first of which was just actually treated last week. That said, unlike the adult transplantation cells that have occurred in the past, these transplantation experiments were done about 10 years ago, the ability of our embryonic stem cell derived retinal pigment epithelium are very robust. Their ability to produce phagocytosis or waste removal, as well as photopigment recycling, are shown to be identical to that in healthy young eyes. So the factors that they're expressing, the potency of these cells, gives us a lot of confidence that as we begin to treat earlier and earlier stage patients, we'll be seeing a much more robust visual acuity results. We weren't looking for visual acuity results in treating these patients. We were looking for no safety signals, which we've not observed any safety signals, but also we were looking for potential surrogate kind of efficacy endpoints. Are the cells engrafting? Are they pigmenting? Is there evidence of photopigment recycling and phagocytosis? And those sorts of things. And those sorts of things that we've been able to observe, we've been able to see in a lot of the patients. And so that's something that we're very pleased with. And we've actually published some photography, both some OCT and some fundus photography, that shows the results of these cells engrafting, surviving, and showing their pigmentation.
So Dr. Del Priore, let me go back to you because you were right. That was the Lancet paper was the less publicly available information. There was both a cessation of loss as well as an improvement in acuity. When we're evaluating companies, when we're evaluating these types of therapies, should we be seeing both? Is it good enough that we just see one? What do you think of these results?
Well, I think that is the microphone on?
Yep.
Okay, I think that the published results are exciting and interesting for a couple of different reasons. So the first thing is it's a phase 1 trial and it's a safety study. So the fact that you've done something in a patient that really hasn't been done before with injecting stem cells into the eye and did not have a serious complication in the first 2 patients and it was reported in a major medical journal is a big issue because there were all sorts of things that could have occurred that did not. So that's the first thing. You've gotten sort of past the safety hurdle. The second thing is that when you're dealing with 2 patients, you're really dealing with 2 patients. So it's very difficult to extrapolate to large numbers of patients. But a visual acuity improvement that was reported in the paper, you have to look at larger number of patients and make sure it's reproducible. It's certainly better than trying to explain a visual acuity decline of the same magnitude in those patients. But you have to take it into perspective. It's a small number of patients. It's a safety trial. The first studies will be safety trials, and the purpose of them is to get you to the next stage of clinical trials.
So let me switch gears real quick to another indication, one that has no real therapeutic options again, spinal cord injury. And Dr. Anderson, this is a field that you have been working in, obviously, for a significant amount of time and have seen the advances that have been taking place from a point of view of timing and dosing and what sort of patients to select. Can you give us an update as to where we are right now in our understanding of spinal cord injury? And there's several companies that are in clinical trials. Just give us an overview of kind of where we've been with cell therapy and where we're going.
Okay, so spinal cord injury, focusing specifically on cell therapy, is really at very early days. And that's true for everything about spinal cord injury. Unlike, well, there are a lot of similarities to stroke or traumatic brain injury where there's a large number of clinical trials that have failed to translate well out of animal studies into the actual clinical setting. We're really at the start of that in the context of spinal cord injury. We don't have this large body of trials that have failed because we're really at the point where we've just gotten there in terms of being able to go forward. So spinal cord injury right now in terms of standard of care is really all about stabilization. So these are by and large traumatic injuries, car accidents, diving accidents, this kind of thing. You have to go in first and foremost, stabilize the spinal cord. We know now out of a set of clinical trials that have been done, the faster you do that, the better the long-term prognosis in terms of spontaneous recovery. There's a lot of spontaneous recovery early. This stabilizes over time. And so what is starting to change, as is the case for stroke, I think, is the very early shots across the bow for spinal cord injury. We're focused on acute therapeutics, things that would protect the central nervous system and keep more damage from happening. Those have not been home runs, but I think we know for the central nervous system, nothing's probably going to be a home run. These are all going to be combinatorial therapeutics that we're looking at in the long run. The shift now is to look at a more stable baseline in terms of focus, right? Very much being able to look at patients who have stabilized over time, use them potentially as their own baseline or in randomized control groups, and being able to see, especially with cell therapeutics, whether or not you can shift that potential for recovery when you're doing a trial perhaps focused on a smaller number of patients that are just very well characterized. And in the context of stem cell treatments or regenerative medicine treatments, I think this is very much the way to go in terms of the future, to enroll a sufficient number of spinal cord injury patients acutely after injury and effectively get a cell therapy on board. While I think Geron showed in their early trial with OPCs that one could potentially get in that direction, it was always going to take enrollment of several hundred subjects, as many as 600 subjects, based on the ICCP historical analysis of the SciGen clinical trials and so on, in order to be able to have a potential to see something that was a clinical effect in a proper randomized trial. Moving towards a more chronic stage and moving in particular into cervical spinal cord injury where we just have better outcome measures, right? Because getting this back is an enormous improvement for somebody who's completely paralyzed, lost all sensory and motor below the level of injury. We can measure that. And we know that it has an enormous impact on patients in their lives and their ability to reintegrate functionally in society, even though it's a very small change to be aspiring to get.
So when we just, I think Jeff Jonas had brought up the fact that it's not about the science anymore, right? And there are other things to be thinking about. But still, we're pretty, early on, in these days, especially in CNS disorders. So tell us just a little bit about the science here. I know from Gil's point of view, it was we're looking more at trophic factors, right, and the trophic effect of these cells and being able to recruit multiple cells. From Gary's point of view, I think there's a lot more integration and potentially replacement. What's happening here in spinal cord?
Sure. It depends what cell you're talking about, right? So lots of things go wrong in spinal cord injury. There's the initial trauma, cells die. That's where neuroprotection comes in terms of stabilizing. But there are some other factors that are very, very important. Over time, the central nervous system builds up a scar, and that prevents reconnection of fibers that used to be connected. And you have to have those signals going up and down in order to have function. So there are therapies that look at breaking down scar, things that look at trying to promote regeneration, promote that reconnection. In the case of stem cell therapies, I think they really come in with possibly a multimodal mechanism of action. There are some types of things, bone marrow stromal cells, mesenchymal cells, for example, which are all cells condition their environment. Those cells make a lot of trophic factors. In fact, most of, for example, the Geron data with OPCs probably supports a neurotrophic mechanism of action, that those cells come in and condition that environment and just make it a friendlier space. The opposite effect that you can have is to actually integrate new cells and get recovery as a result. In spinal cord injury, a particular issue has to do with the loss of insulation around the fibers that send messages up and down. And that insulation in the central nervous system is called myelin. And it's lost after spinal cord injury because you lose a population of cells that are called oligodendrocytes. And so in at least some cases, the path that's been followed is to try and see if you can get a population of cells to permanently integrate. So this is Stem Cell Inc.'s strategy, really, in terms of where things have gone, showing that those cells are capable of remyelinating, replacing that insulation, and improving repair by that mechanism. But I think stem cell therapies, really, it's a cell-by-cell specific case. You might get neuroprotection in one case, neurotrophic recovery or promotion of regeneration in another, and cell replacement that gives you function depending upon the cell type that you're thinking about applying. What the mechanism of action is, therefore, is really important to understand in terms of patient selection, in terms of patient stratification, in terms of what's going to let you be able to see in a clinical setting, did you make people better or did you make them worse?
So when we talk about, like you mentioned, there are several companies that are in clinical trials right now. There are several investors that are here that are probably never going to invest, let's say, in preclinical studies, right, or data that's coming from preclinical studies, which is one of the questions that we have at the end, but only after clinical studies. But help us to think about what we need to see to have, to potentially invest in a viable therapeutic. So, like you said, this is a big deal for someone who is a paraplegic, right? But is that measurable? I mean, when you talk about the endpoints that get you excited, what are they? And you also mentioned spontaneous recovery, right? So, when we see someone, having that kind of a recovery right away, what are the questions we should ask to know if that's spontaneous or due to the cells?
So sort of like stroke, there's a lot of accumulated knowledge about what the kind of natural history of disease is for spinal cord injury. So we know in people, and there are reviews and multi-center trial analyses that are published on this, showing that most of the spontaneous recovery that's going to happen is within the first 3 months post-injury. Continued recovery out through 6 months. By the time you hit 12 months, you're in subjects that are at a very stable baseline in terms of where they're going to be for function for the rest of their lives. What do you need to see either in a preclinical model or in a clinical trial to make you say sort of wow, I think is what you're asking. That's a good effect. I think to put things in perspective is to understand what the deficit really is for people that are living with this, right? There are very few indications in terms of neurological injury or disease where we say you have no sensory or motor function at all and you should be happy with that, right? This is an enormous impact for people's lives. And I think one of the things that doesn't line up well is patients' perception of what they would like to get back, physicians' perceptions of what they would like people to get back, and the sorts of stuff that we look at in the lab. And none of these three things have often been in very good alignment. For individuals living with spinal cord injury, I think it's really important to understand the context of what deficits they're living with. These people can't control bladder function, bowel function. They've lost sensation below the level of injury. They don't know if they gashed their leg, and they have a pressure sore, which could be life-threatening. They develop complications like autonomic dysreflexia, which is a dysregulation of things like being able to control your blood pressure. So that if I get stuck in the traffic of LA for 2 hours, it's a huge inconvenience, and I'll be very upset about it. If you have spinal cord injury and you're lucky enough to be functional, to have a car that you can drive, this is potentially life-threatening because they can't automatically cath themselves to empty their bladder, and it could trigger an autonomic dysreflexive episode that causes them to stroke out and worsen their injury, right? So what people with spinal cord injury want to have back is bladder function. Number one on their list, if you survey. What they'd like to have is bowel function. What they'd like to do is not have to take 4 hours of their day and take care of those functions manually or with an assistant, because that keeps them out of the workforce. It keeps them from picking up their kids. And the consequence of that in the context of clinical translation is that the higher up the injury and the longer term the injury, this costs a lot of money. So while we have about 1.3 million people living with spinal cord injury in the US today, in 1998 the cost of medical care for that 1 million people is over $40 billion. So if you do the math and calculate that out, it's disproportionately large, right? So something that can give bladder function back and prevent urinary tract infections, prevent autonomic dysreflexic episodes in the clinical population is an enormous impact, right? I'm excited about bladder function. But the problem is that many people would look at this, certainly in the lab, and think you don't have a HITS unless you have an animal that's walking perfectly normally. The clinical endpoint that might be logical is how fast somebody can walk across the room. That's a HITS. That's a great home run. But it is not the most important part of the market, in my opinion.
So maybe we can switch gears to Steve very quickly, and you can talk a little bit about where StemCells Inc. is at in the clinic, and just incorporating what Dr. Anderson talked about as far as these other endpoints that might be more important for these patients, and related to that, probably even more importantly, is what's the regulatory agency saying about those endpoints? Because again, going back to earlier talks, the keynote talks, this is a revolutionary industry, and it seems like you need to come up with newer methods of really understanding the benefits.
So lots of good points. You're right. This is a revolutionary industry in the sense that in collaboration with scientists like Aileen and clinician investigators and companies, we're trying to create a field in terms of how you might apply stem cell therapeutic to central nervous system disorders, and that in and of itself has a lot of challenges. At StemCells Inc., the company is a publicly traded company, and we're developing a purified population of neural stem cells, and to see what their therapeutic application is the lead product for the company for a wide range of neurological disorders. I think you can get from the flavor of what we're discussing versus our colleagues in cardiac that the CNS is going to develop with a different feel. It's going to be fewer patients, but our signals that we'll get from the trial will be different. And maybe there'll be things that we don't predict, and there may be things that are important to the patient, but not necessarily what we see in preclinical experiments. So as we develop the field, we have to be sensitive to all those things. At StemCells Inc., one of the ways that we've thought about this is to very much follow the science. So take the data that we can get from preclinical studies, and then and selectively choose which of those make sense to begin human testing. And we're feeling our way forward. But the good thing is we are advancing. We've now completed 2 trials for brain indications. One, a lysosomal storage disease, so a diffuse central nervous system disorder, and another for a congenital myelination problem, both of those in children. Both trials are completed. And the good thing is for the field is that up now to 1 billion cells have been placed into the brain of children with the diffuse disorder. And now those children are 5 years out from their surgery, and we have no safety concerns. So that's an important milestone for the field. The next trial that we did, to put it in perspective, we now start to see evidence of biological activity, and I'm referring to the fact that when we put the cells into an environment that it is missing the insulation, this dysmyelination syndrome, we see signals by MRI and perhaps clinically of some type of impact of the cell. In terms of spinal cord injury, the trial that we're conducting now, which is the field starting to advance, we're starting, in my view, a good thing as a neurosurgeon, we're starting to ask these questions. We're going to get clarity if there's a value to these particular cells. The phase 1/2 trial we have now underway at the University of Zurich is going to be looking at patients who are in the chronic stage of disease. So we have a background from which we can tell, is there something that looks like it's a deflection from the natural history of the disease? We're also, I think, important for this field and across all diagnoses is what is the optimal patient population to first test the cells in? Now, we all recognize the risk-benefit of going into patients who have the least to lose as you begin, but I think we also have to acknowledge that we've got to test this product as early as we can, these cells, in patients that have a more reasonable chance of some kind of clinical benefit where we can detect a signal. So that it means being very aggressive with our first initial trials because we're not going to get a lot of swings at the bat. We understand that. So we're not going to have hundreds of patients in our early trials, but what we're going to have is fewer patients, more intensely studied with a dose that we try to scale up from an animal setting that we think might be therapeutic, with endpoints that have a mixture of both surrogate, which is a predictor of a biological effect, as well as a direct clinical impact. Again, speaking as a neurosurgeon, I come at this more with my heart and my mind than I do from a scientific standpoint, and I recognize that there's a huge unmet need. I've talked to patients who have spinal cord injury, and at the end of their acute care, when you stabilize them, it's very disheartening to say there's nothing else we can offer. We're not at that stage anymore. We're beginning to ask very good questions with very good cell types, and I think we're going to advance. And in particular, from a perspective of a clinician, if you look at the properties of a stem cell, the inherent biological properties of it's multipotent, so it can make all the different cells in the brain. It can migrate within the brain. It can self-renew. If you begin to add all those up, you begin to think as a therapeutic, that has the potential for a groundbreaking achievement in medicine, because it's a one-time intervention that could address perhaps more than one central nervous system disorder. And a little bit, as Aileen said, will go a long way as we begin.
So let me ask you just to follow up, what has been the discussion with the regulatory agencies in, when designing these trials and looking at these other, are they amenable to these other endpoints? Are they kind of, hard stuck with their known endpoints?
I think all of us here in this room have had interactions with regulatory bodies. I'm encouraged by the fact that I think the regulatory bodies are very interested in this field. I think they are willing to engage in a dialogue about how we can look at this. I think the agencies really understand that there has to be a more openness to letting these interventions advance, and advance in an aggressive way, balancing, of course, safety. So my sense is that the agencies are very much open to a dialogue, and if we propose something that's rational, that's justified, that has a risk-benefit, which is where their central point of focus is, that makes sense to them, that's how we'll advance. Now, that all takes a lot of thoughtfulness, but I think we definitely have now a team of investigators that we can accomplish that with.
Can I just comment?
You bet, please.
So one of the things that I think is really worth understanding about the field and the community of a lot of different companies and researchers that are here today is that over the past several years, we've actually made a proactive effort through the Alliance for Regenerative Medicine and kind of the broader community to engage with the FDA and other regulators around the world so that we could actually take on some of the historical challenges that relate to a perceived lack of consistency or a lack of support or a lack of willingness to engage around some of the thorny regulatory issues that many people feel have been holding back the field. And what we've learned is that if you approach the FDA and other regulators in a way that is positive, constructive, open-minded, and you're willing to engage in a true dialogue with them, then you're going to get a lot more out of that interaction. And I think just last year, for example, when there was some pretty important regulatory changes that went into effect as part of PDUFA 5, the Prescription Drug User Fee Act, kind of the fifth iteration of it, if you will, ARM and Bio and other people that were involved in that process got some very substantial changes put into effect that relate to things like a broadening of the accelerated approval pathway, the designation or the creation, if you will, of a breakthrough therapies paradigm. The FDA is talking about other changes which could actually even accelerate even further these types of therapies in a way that make it more realistic for people to go after some of the major challenges that, frankly, traditional medicine has been completely unwilling or unable to have an impact in. That is incredibly exciting for anybody that's really interested in the field of stem cell therapy and regenerative medicine as a whole, because it means that there are efficient or more efficient pathways forward that we as sponsors or innovators, as clinicians, as patients, and as investors can take advantage of so that we can actually turn these things into a medical reality and do it in a scalable, reproducible, consistent way.
And I think that investors should be able to appreciate that right now because of this, the whole advent of breakthrough therapies, breakthrough designations. We've always had accelerated approval and the like, but clearly there does seem to be a willingness to work earlier and to move forward with really high potent therapeutics that seem to change the trajectory of a disease. I'm sorry, Dr. Anderson.
So just in this context of whether how those alternative endpoints are perceived, right, to different endpoints. I think certainly in stroke and spinal cord injury, most regulatory bodies, the signals that I see are listening to the field, right? And there's been a lot of grappling by basic scientists, by clinicians in the field with what should we be looking at for endpoints. Spinal cord injury is a really good example of that, where there's been really a concerted effort over the last 10 or 11 years by a number of nonprofit groups, ICCP and others, to say, we're getting close where we can go to good trials, and we don't have good enough endpoints. And so in advance of that, there's been a lot of work back at the interface of the basic science and where it meets clinic, and looking at what can we do well in animal models that picks up factors that we really think are going to be important in the clinic, and then how can we test that in a reliable way? How can we test hand function? How can we test recovery of sensory function, which might be very, very critical for spinal cord injury, and mirror what we see in animal models to what we see in the clinic? And so I think on a positive side, from a regulatory perspective, that input in that period of time in terms of development is being listened to.
I mean, for the ophthalmology indications, it's easy to test for things, but it's much harder to decide what it is you're really looking for. What kind of visual acuity improvement or stemming the decline of the disease are you really trying to identify? And over what period of time do you have to look at it? So it's interesting how you look at the different diseases and what the endpoints are and how you set them.
Yeah, one of the common themes that we're all dealing with and having discussions with regulators around it and others is how do you quantify improvements in quality of life, right? They're the easy things, right? If the patient gets up out of the wheelchair and starts walking across the room, you say, hallelujah, we have a success, right? Or for a stroke patient that was so debilitated that they have no hope of actually having a reasonable quality of life. Okay, those are the easy victories, but there's a lot of shades of gray in this conversation. And I think that both for the regulators as well as for the third-party payers, being able to make a clear argument about, hey, here's how we quantify the impact we can achieve, and here's how that translates to value. The interesting thing is, as Aileen was mentioning, if you look at the economic impact for all of these conditions, it's absolutely enormous, and for many of them it goes on for many, many, many years, right? So if we can demonstrate quality of life and significant improvement, not that in many cases it doesn't just touch the patient, it touches the families of the patients that are called upon to deliver a lot of this care, and then make a compelling value argument to the third-party payers, well, that translates into what could be some of the biggest home runs or transformational opportunities in medicine as we know it.
Dr. Wechsler.
So, yeah, I was just going to make a similar point to what Gil just brought out, which is that in my interactions with regulatory agencies, FDA, I think they understand what we understand, that these endpoints are not adequate. It's just that it's the best one that they have right now. And I think it's our failure to not be able to bring to them better endpoints that we can show them and convince them are just as good or better than the ones they're currently using that is keeping things from moving forward. I think they are completely open to looking at new endpoints, and as I say, I think that they understand the deficiencies. But we as a field, all of us in this room and everyone in academia needs to think through and find the better endpoints and give them compelling arguments to why they should use those endpoints as opposed to what they're using now. If we can do that, I think then it will move to where we want it to go.
So I know that we've just scratched the surface on a lot of different indications, and so there's a lot more questions. But I also understand that we and my distinguished panel are all that blocks you between you and a fine lunch. But I would like to push the lunch off for a couple of minutes to just at least give the audience a chance to ask any questions that you may have, because I have a ton, but I won't prevent you from eating food. Any questions in the audience? Yes.
Hi, is this on? Hi, I'm Rahul Saragasar at the Center for Commercialization of Regenerative Medicine, say that 5 times fast, in Toronto, Ontario, Canada. We're a translation center. And my question is about cells as tools. And earlier Kieran Murphy talked about cardiac cells and being, and the cardiac field being about 5 years ahead of the neuro field, and the need for assays in vitro assays to be able to test small molecule and large molecule drugs. And I know that a lot of the conversation has been around the regulatory, but I'd appreciate some comments around the need for these assays in the field.
Sure.
Yeah, well, it's critical. In fact, one of the things that we've been doing as part of the team of leadership from the Alliance over the past couple of years is engage with the FDA exactly on that issue, which is to how do you create a consistent framework so that you can ultimately develop potency assays, right? And to develop potency assays around cell-based products, which are almost by definition, they're multimodal, right? We've talked about this over and over again through the course of the panels this morning. They're not just going to do one thing, they're going to do several different things. How do you quantify that, and how do you do it in a consistent way so you can be sure that your product or your procedure, right, is going to give you a reliable result? And the bad news is that there's no simple solution for that, right? It's not just do A, do B, and do C, and then you're done. The good news is that the FDA is fully engaged in that conversation, as are other regulators, to work with us as innovators, clinicians, and sponsors to try and figure out, well, how do we do that more effectively? Now, in fact, the FDA just recently, just in January, had a conference as part of a workshop on this to focus on those specific issues. And The really exciting thing is that in the last few years, we've actually discovered that we can do things like take human cells and put them into animal models, and then in very sophisticated ways using gene chip technology and other new assays and technologies that have become available just in the last decade, that we can actually begin to precisely measure and quantify what the biological effects of that are. Well, that ultimately leads you to a point where you can say, hey, these are the key pathways and these are the key factors that we're regulating, and now here's how we actually take that information and turn it into potency assays. But ultimately, in terms of large-scale application, if you want to make this standard of care, well, you've got to be prepared to think about how are we going to take that information and turn it into potency assays so that we can show that our product does what we think it does and does it reliably.
I'll just add that, I mean, there's a range of different cell types. I mean, it really just depends on your mechanism of action, or if there are multiple different mechanisms, at least if you could delve into trying to tease at least one or two of them out, and there are various different assays that could be used. So if you think that your approach actually could be stimulating neurogenesis, then obviously there are really interesting approaches that have come down the road, which you could actually use human neural stem cells, for example, and see whether they get stimulated or not by your cell therapy approach. Just as one example, or you could have angiogenesis looked at in vitro. Cytomedics has assays around things of that sort. So there are various different approaches. The other way to look at this is also because I was seeing that first lecture as well around the beating cardiomyocytes. You can look at toxicity. So another thing, of course, that's come down the pathway is iPS technologies, where you can grow out skin cells from patients with Huntington's disease, for example, or Parkinson's disease, and turn them into neurons, and you have a wonderful opportunity to look at assays for toxicity and drug screening and so forth.
Yep.
Just a very quick question. How long do stem cells that are injected into various areas, how long do they survive? Do they split and become progenitor cells? Do they develop other cells? Have you been able to track the life on those?
So if you look at the data that we've got so far with regard to neural stem cell transplantation directly into the brain that we can derive from post-mortem studies from patients who have died from complications related to their underlying disease, what we've shown in the NCL trial, which was the lysosomal storage disease trial, is that the cells survive out to 2.5 years after transplantation and 1.5 years after we stopped immunosuppression, the planned cessation of immunosuppression. So we're starting to see the same kind of effects in terms of cell survival in the human condition that we've observed for long-term survival in the animal model. So it's encouraging to us that we can demonstrate this long-term survival, because I think part of the effect is going to be long-term engraftment of the cell.
In the case of our cells, our earliest patients were treated 18 months ago, and we have some quite nice pictures of those cells continuing to survive and persist 18 months later. This is actually longer than the life of animal studies we did. The life of the animals that we did with the rats and mice were only about 1 year. So we're seeing survival and engraftment and persistence of these cells 18 months in.
Yeah, sorry, go ahead.
And do you use immunotherapy as well in addition to that?
We initially treat patients with a course of immune suppression, but they're weaned off of it at some point between 6 weeks and 6 months after the surgery. So.
Well, I was going to say, and other cell types may have more of a drug or a biologics kind of profile. So what we've seen with multistem over and over again is that the cells don't permanently engraft, which is actually in contrast to what we thought they may do when we first started working with them over a decade ago. What we've seen is they like to home to sites of tissue damage and inflammation, or to certain types of areas of injury. And then when they get there, they get busy and they interact with a bunch of other cells, but within a few weeks, they're all gone. So it creates different options and different, and thinking kind of 2 steps further out, you can envision a day that some of these things may actually work very synergistically, right? Cells that are designed to be around for a long time and cells that are not. You may see some very powerful effects by combining some of those platforms.
So let me wrap this up with the central theme, I think, in regenerative medicine being more a restoration of balance, and I hope that we can start to end the debate between trophic factors and replacement mechanisms of action, but rather, a combination of factors that are frankly beginning to restore the balance in the body after injury. So with that, I want to thank the panel for their participation here.